Hi guys, welcome back to the PhD Prep Lab channel. We have yet another video for you guys for the quick revision series, one shot video for the Part B ICMR JRF examination. So today we are going to cover yet another very important unit from the Part B of ICMR JRF examination syllabus, which is the common bacterial, viral, fungal, and parasitic infections. We often tend to overlook this unit sometimes because of maybe lack of concise and crisp notes. So here is your chance. This is a one shot video. We have this quick revision video and a crisp form of notes which you can get on all these bacterial, viral, fungal, and parasitic infections, which is very important. So make sure that you don't miss it out and you do revise it. Do watch this video before your ICMR examination. So before we move on to the video, I would like to remind you that we have uploaded other one-shot videos from the part B of the ICMR examination, ICMR JRF examination. So we have one-shot videos on genetics and genomics, evolution, then on animal physiology, basic animal physiology, then biosafety, bioethics, etc. So you can check out the links from our description box. So make sure that you have covered those videos as well, and then you watch this video as well. So let's start the video before delaying. So let's talk about the basics of the bacterial cell. We all know that it is a prokaryotic type of cell, so we don't see any defined nucleus or nor there is any membrane bound organelle present. So it is very important to know which organelles are membrane bound and which are not, because these questions are often asked. They give you four organelles, four types of cell organelles, and then they ask you which will be absent or present in prokaryotic or eukaryotic cell, depending on the question. So these questions are often seen in ICMR, DVT, even in your CSIR JRF examinations. So this point is very important. And they are very small, usually from 0.2 to 2 micrometer in size. They have a cell membrane which is surrounded by a cell wall made of peptidoglycan. So their cell wall composed of peptidoglycan, and in gram-positive bacteria, we can see a thick layer of peptidoglycan, whereas in gram-negative, there is a very thin wall of peptidoglycan. An important point to note that gram-negative bacteria, it has lipid as a part of its outer membrane, which also acts as an endotoxin. So this is also one very important point. Now we have two types of bacterial stains, which is first the gram-positive one and then the gram-negative one. So it is important to also remember the stain color. So the gram-positive bacteria, they stain blue or purple in the presence of gram stain, whereas the gram-negative bacteria, they stain red in color. And mycoplasma cannot be stained because they don't have any cell wall. Now in the other column, you can see the shapes of the bacteria, which are usually present. You see cocci, which is spherical shape, bacilli, which is rod shaped, etc. like that. Then another note on that mycoplasma, they don't have any cell wall. And then you have bacteria depending on the use or not using the oxygen. So you have aerobic bacteria. First, it would be obligate aerobes, which means they require oxygen 100%. And then you have anaerobic bacteria as well, which could be obligate anaerobes, means they cannot live in oxygen at all. Or you could have facultative anaerobes, which means they can grow with or without oxygen. Now you have another category as well, which is microaerophilic, which means they require low oxygen and use fermentation for energy. So this is also an important group of bacteria, microaerophilic. So now we'll be discussing some diseases which are caused by bacteria. So first we have tuberculosis to discuss. So this is also known as TB. So this bacteria, which is Microbacterium tuberculosis, it is acid-fast bacteria which has a lipid-rich cell wall, and the transmission of this disease is usually through airborne particles from the infected individuals. And there are certain stages of TB. So first, it happens when an infected person he sneezes, and then through the aerosol particles, the Microbacterium tuberculosis stains, they are gone inside the body by inhalation or some other manner in the healthy individual, where it goes first in the alveoli of the lungs. So from there, what happens to the microbacteria, the Microbacterium tuberculosis bacteria, is a series of events that follows. So after the bacteria reaches the alveoli, it generates an immune response. Firstly, the innate immune response, wherein it calls out the macrophages. So there is recruitment of new macrophages and multiplication and spread of the bacteria therein, which expands the infection. And more immune cells are also arrived later, which includes the lymphocytes and the neutrophil to the infection site. So after this happens, there is a structure formed which is called as a granuloma, which is a very important structure in this whole life cycle of the Mycobacterium tuberculosis. So this whole structure of granuloma, it contains of certain components. which includes the T cells, B cells, fibroblasts, dendritic cells, the natural killer cells, the neutrophils, and also the active bacteria also. So this granuloma structure basically it suppresses the spreading of the TB disease. So in this stage, the bacteria is dormant in nature. It does not allow this granuloma structure does not allow the TB bacteria to break through and spread to the entire body. So at this stage, the host and the infected person, the host here is asymptomatic in nature. So here the immune system is acting heavy on the bacteria. But due to certain immunological conditions, when your immune system is compromised, it could be because of anything. So then the bacteria overpowers and it takes over your immune system, wherein the granuloma disintegrates and the bacteria they break through and they spread across your whole body and various symptoms of the TB can then be seen. So it is also called the symptomatic stage here. So this, when this happens, so then you can detect the TB in the host or the host person. So this marks the spreading of the TB in your body. So you can see certain um, symptoms as well, which marks cough and hemoptysis, like when it touch your cough will contain blood. You also observe weight loss, night sweats, etc. So for the diagnosis, usually the screening is done through PPD skin test or IGRA, which is a blood test, wherein your imaging of the X-ray is also done in the imaging section. And then to confirm all this, the diagnosis, the confirmation is the sputum culture, which is the gold standard for diagnosis TB, tuberculosis. And then for treatment, you have RIPE therapy for 2 months. And the composition of RIPE is mentioned here on the screen, you can see. And then you have wherein you have latent TB, so a person with latent TB is usually given ionized monotherapy for 9 months. The other disease that we have here is typhoid. So this is a kind of fever. This happens due to a gram-negative type of bacteria, which is Salmonella typhi. And the transmission of this disease is usually via the fecal-oral route. And firstly, you get it through the contaminated food and water. So what happens is when a healthy person ingests a food, any food or water which is contaminated in nature with this uh, typhoid bacteria, which is Salmonella typhi. So the bacteria it goes inside the host body, the healthy host body, and then after passing the normal digestive system it goes into the intestine, and then there it starts its colonization, especially in the ileum of the small intestine and the Peyer's patches. So there the bacteria adheres and it sort of forms an attachment, and biofilm formation is seen for long-term persistence of the bacteria. Then through the intestine, the bacteria travels and it reaches the enterocytes lining the terminal ileum of the intestine. And then from there, it passes the Peyer's patches, and from there, it follows a route of the lymph nodes, wherein this is in the form of the primary bacterioma, which is usually asymptomatic in nature. And even if you draw the blood of the person and you test it, the test usually comes out to be negative for typhoid. So after some time, the bacteria develops, the primary bacteriema is developing, and the infected bacteria it reaches the liver, spleen, and the bone marrow. So after it reaches these places, then there is extensive multiplication and colonization of these bacteria into these organs, as I mentioned, like your liver, spleen, and bone marrow. So there they colonize and they multiply, wherein they may replicate within the macrophages as well. So here they show the clinical symptoms. The host will show the clinical symptoms because now it has become the secondary bacteremia. So here you will get symptoms like your fever and headache and other constipation related a fever, because here the continuous shedding of the bacteria is happening into the intestine, and the feces also they have the strains of the bacteria. So fecal test also tests positive for typhoid here. The fever that occurs in typhoid is mainly due to the endotoxin and the cytokines. The endotoxin here is the LPS. So the diagnosis is done via the blood and your stool or urine culture. So initially the test is done on the blood, and then later on the test is conducted on stool and urine as well. And the most important and widely famous test here for typhoid is the Vidal test. And some drugs that are mentioned, the first line of treatment is it could be azithromycin or ceftriaxone. The alternative treatments is also mentioned like ciprofloxacin. Then in severe cases, meropenem is given. and this is how your typhoid gets treated over time. Then we have cholera, which is caused by Vibrio cholerae, which is again a gram-negative bacteria. It is comma-shaped, and the transmission is again here through fecal-oral route and spreads. You first get it from the contaminated food, water, seafood, shellfish, certain things like that. And here it produces the cholera bacteria produces a toxin known as choleragen, and it activates the G protein, and there is massive secretion of chlorine, sodium, potassium, ions. The first hallmark of the symptom of cholera is the rice water like diarrhea, which is very, very watery in nature. And the stools are not bloody, they are just very, very watery. Other signs will be your vomiting, cramps, loss of electrolytes, and then severe complications could be your dehydration or even circulatory collapse. And no abdominal cramps, fever, or pain is typically observed here in case of cholera. The diagnosis is usually done through stool culture, and a very famous test here, which is used, is the dipstick test. And the molecular diagnosis could be done through PCR as well. And mainly, the treatment includes fluid and electrolyte replacement or certain antibiotics, wherein the doxycycline or azithromycin is used. Or the second choice would be TMP, SMX, or ciprofloxacin. Now we have another disease which is by a gram-positive bacteria. So this is tetanus, and this is mainly like the lockjaw disease, which you can say, and it basically is due to the secretion of the neurotoxin, which blocks the inhibitory neurotransmitters GABA and glycine, which is basically it results in the uncontrolled muscle contraction. So the important point that you have to remember here is that tetanus is also called the lockjaw disease. So wherein also the other important point is the neurotoxin which blocks GABA and glycine. So it mainly you get it when you get a very deep cut through any contaminated surface. It could be a road or a knife or any rusted surface. And when you get a deep wound and you don't treat it, you don't get the anti-tetanus shots from the pharmacy. So then there are high chances that you are contaminated, like your blood has been contaminated by the Clostridium tetani bacteria, and you may develop this very deadly disease. So it does lead to death, and it's a very pathetic disorder which gives you uncontrollable muscle spasms. You could die even on choking on the food because all the muscles of your body they are uncontrollably just contracting, and that's how usually the death occurs. The diagnosis could be entirely based on when you got the wound or the injury, or what are your symptoms, like which kind of spasms you are getting, or stiffness in the body you're getting, or sometimes in certain conditions the body also becomes arrow shaped, like there is arcing of the body. So it's a very deadly disorder if you ever get a wound from a contaminated space, or if you have a doubt whether the space was contaminated or not, it's better to get an anti-tetanus shot than to regret things later. So the treatment includes the human tetanus immune globulin HTIG, which is to be given immediately, certain antibiotics, and other things like you get a booster of tetanus shot every 10 years. So these are some preventions that you can follow. The next disease here is botulism, which is caused by Clostridium botulinum, which is a gram-positive bacteria. You usually get it through poor quality foods like your canned foods, where which does not survive poor heating, or you get it from contaminated soil or raw honey in case of infants. And it also produces a neurotoxin, which is very lethal in nature, which basically cleaves the SNARC proteins, which blocks acetylcholine release at the neurotransmission junction, which causes flaccid paralysis. There are certain symptoms of this, which includes the classical, like your 4 Ds of botulism, which includes diplopia, which is the double vision due to paralysis of ocular muscles. Then you get dysphagia, which is difficulty in swallowing. Then you get dysarthria as well, which is difficulty in speaking due to the weakness of tongue and facial muscles. And then you also see dysphonia, which refers to a disorder of the voice production because of the complete loss of voice and paralysis of the larynx, the vocal cords. So the 4 Ds are important, and you also see dry mouth and muscle weakness, which includes your respiratory failure as well. So in infants, this disease is called the floppy baby syndrome. So this floppy baby term is important, could be as a one-liner, like a very short type of question in MCQ form. The diagnosis includes your symptoms and history, and the confirmation is to detect the toxin in the serum, your blood serum, stool, or food source. And then you have some treatment options here, which includes antitoxin, HBAP in adults or baby pig in infants. Then you have wound botulism, which includes the surgical debridement along with some antibiotics like penicillin. Now, here are the list of gram-positive bacteria along with the diseases they cause. So first you have Bacillus anthracis, which causes anthrax. Then you have Bacillus cereus, which causes food poisoning. Then you have Listeria monocytogenes, which causes meningitis. And then Corynebacterium diphtheria, which is causing the diphtheria or the bull neck. Then you have anaerobic bacteria, all under gram-positive here. So you have Clostridium botulinum, like we discussed, it causes the botulism. Then you have Clostridium tetani, Clostridium perfringens, Clostridium difficile, which causes antibiotic-associated diarrhea. Then you have Actinomyces bacteria as well, which causes the sulfur granules. Now we have gram-negative bacteria, which causes the certain type of disease like Neisseria meningitis, Neisseria gonorrhoeae, which is an STD, causes an STD. Then we have Moraxella catarrhalis, which causes otitis media in kids. Then you have E. coli, which is a number of one cause of the UTI or bloody diarrhea or HUS kidney failures. You have Salmonella proteus, which also again causes UTI. You have Vibrio cholerae, which causes cholera, the rice water stools. Then you have Helicobacter pylori, which causes peptic ulcers, gastric ulcers, and other diseases. So it's important that you know all the bacteria and which diseases they cause. So it's very, very important. One or two questions will definitely come from this. Now you have certain respiratory and zoonotic disorders. The zoonotic disorders is the disorders that we get through the animals. So here are some lists which includes your Haemophilus influenzae, your Bordetella pertussis, which is your whooping cough, Yersinia pestis, which causes plaque. Then you have Pasteurella, which you get from the dog or the cat bites. Then you have Treponema pallidum, which causes syphilis. Then you have chlamydia, which also is number one bacterial STD disorder. So you have these disorders and the causative agents from where you get. So it's also important that these are like the most common and from which questions have already been asked and are usually seen in number of JRF examinations. So these are like this is an important list that you must go through. Now there are different classes of antibiotics, and this is an overview for you to look at. So I'll not spend much time on it. This is pretty self-explanatory. I want you to sit and relax and have a look at it, and then go through one by one, go through each antibiotics one by one. It will be very helpful for you for the revision series as a for your ICMR examination. So make sure that you have a look at it. You can like get the notes from our Telegram channel. You will get the scanner at the end of the video. So make sure that you join that group and therein you can get the notes of all the one-shot videos, including this as well. Okay, so let's start with the viruses now. So we'll discuss the common infections that are caused by viruses and the classification of viruses as well. To start with, viruses are considered in neither living nor non-living. So they are actually non-living when they live outside a cell, but when they integrate to a host cell, they are considered to be living. They can have a genetic material which could consist of RNA or DNA as well. So it could be either RNA viruses or DNA viruses based upon the genetic material. And their genetic material of the virus are actually surrounded by the protein code, which is called capsid. So capsids could be helical in nature, icosahedral in nature, or complex made up of complex materials. In some viruses, the capsids are surrounded by lipid bilayer membranes, which is derived from the host cell membrane only, and which is actually called an envelope. The envelope could be either naked or it could be like envelope, like cover. So some viruses they have spikes like glycoprotein molecules on their outer membrane, which have functions including the attachment of the virus to the host cell receptor. The average size of a virus could range from 20 to 200 nanometer as well. So we have here the very detailed classification of virus. Now, it is not, so basic idea is to that you remember certain examples of mainly RNA virus and DNA virus, like what comes under which of the viruses are having RNA as their genetic material, which of the viruses they have DNA as their genetic material. So this main important point is necessary. This is a very detailed classification, but for exam point of view, for a quick revision basis, only to know which are RNA viruses and which are DNA viruses is quite sufficient and helpful as well. So we'll discuss here. So viruses are divided into two categories based on their genetic materials. First is the RNA virus. So the RNA in there, or like the genetic material, it could be single stranded as well and could be double stranded as well. And under single stranded, it could be positively stranded or negatively stranded. And under positive stranded, you have envelope, naked, or in negative stranded, you have envelope. So this is the detailed classification. But I'll tell you that RNA viruses, the major names that you should remember is like your SARS-CoV virus, your HIV virus, then your Norwalk virus, rhinovirus, poliovirus. So these are hepatitis A virus. These are important. Then your sandfly fever virus is also RNA virus. Influenza virus is RNA virus. Mumps, measles viruses, and influenza viruses, along with your rabies virus is also RNA virus, and rotavirus is also RNA virus. And your DNA viruses, they include herpes simplex virus, varicella zoster virus, which causes your chickenpox. Then you have smallpox virus, parvovirus, adenovirus, human papilloma virus. So these are your DNA viruses. Now we have the very widely studied viral disorder, which is AIDS, acquired immunodeficiency syndrome. So this is the virus that causes AIDS is actually HIV virus, the human immunodeficiency virus. So this virus mainly targets the immune cells, which has CD4 receptors on their surface, which includes the T cells, dendritic cells, macrophages, and others as well. So HIV is a single-stranded positive sense RNA virus. Mainly to remember is that HIV is a RNA virus and it is encapsulated with a layer with bilayer of lipid membrane. This point is also very important. And on the surface of the enveloped virus, it has a glycoprotein GP120 and GP41, which binds to the CD4 receptors and their co-receptors on the host cell membrane. So the HIV, it enters the host body and it infects the CD4 T cells, macrophages, and dendritic cells. Then with the help of this GP120 and GP41, like with the help of this GP120 glycoprotein, the virus binds to the CD4 receptor and their co-receptors, and the membrane fusion is actually mediated by the GP41 glycoprotein. And after the virus enters the human body, the viral RNA is released and converted into double-stranded DNA by reverse transcriptase. This viral DNA is transported to the nucleus and it's integrated into the host genome by the help of integrase, and which forms actually a provirus inside the host body. So now the host machinery then transcribes this provirus and translates the viral genes. So the new virons, they assemble at the cell membrane, they bud out and they mature, they are released into the host body. So this produces more infectious viral particles, which actually leads to the spreading of the disease to the entire body of the host. So there are two subtypes of HIV, HIV-1 and HIV-2. HIV-1 strain is majorly seen in US and other parts of the world, but HIV-2 is mostly seen in South Africa and south part of Asia. The symptoms are usually like you could be asymptomatic till up to 10 years after you have acquired the AIDS infection. So usually it starts with general symptoms like fever, sore throat, weight loss, fatigue, and over the time gradually increases to severe symptoms. Up to 10 years it would take time, and then is the time when you actually get tested and you test positive, and the major indicator is the extreme reduction in the T cells. So when the T cells number they reduce below 200 or the total T cell percentage reduce below 14% of the total lymphocyte count, then it indicates AIDS. And the patients of AIDS, they have defining illnesses. So a lot of things, their immune system is compromised so much that even minute infections are very easily catched by them, which aggregates to severe infections and problems. So this is the whole stages how the AIDS infection, the HIV infection, it occurs. So HIV infects the mucosa, then it targets the CD4 T cells, it attaches to the dendritic cells, it travels to the lymphoid tissues, it replicates, and infection is established. Then there is HIV spread through the circulation, which is called viremia. Then the immune response, the partial containment of seroconversion is there. And then there's clinical latency, you could be asymptomatic for up to 10 years. Then slowly there is CD4 depletion and immune suppression is also seen. So there is no actual cure of AIDS. It's just that you could suppress the symptoms for a certain amount of time until your whole system gives up. So a table of treatment is also given, which they use NRTI, IMSTs, which certainly stops the integration of the virus and stops building up of the virus. So these are just to mild down the symptoms or the worsening conditions, not to exactly cure it. Then we have influenza and infection. So this is also by the RNA virus. So it belongs to the Orthomyxoviridae family. There are certain types of influenza infection. Type A1, which is most severe and causes the pandemics, and type B is infecting only the humans, and type C is a mild type of influenza. So the common subtypes, it also includes H1N1 and H3N2 strains. The transmission is usually through the respiratory route. It multiplies in the mucosa, causing its inflammation. Some signs and symptoms which are very common include fevers, chills, cough, pain. The children are more prone to it, and the diagnosis, it includes the rapid influenza test or the PCR test. There are certain other types of influenza treatments as well, which is basically rest or some antivirals or some vaccinations are also given just to uh, treat certain types of strains. Now we have another life-threatening viral disorder, which is hepatitis, and it has four types: A, B, C, D, and E. So five types basically. Hepatitis basically infects the hepatocytes of the liver, so basically the liver cells, and it causes severe liver inflammation. And hepatitis B is the most deadly one, so it is also a viral disorder. So this basically in HIV-A we see uh, that it is made up of the RNA genome. The genome here is RNA. Even for hepatitis C, D, and E, the genome is RNA. But in hepatitis D, it is a DNA type of virus. It has a DNA genetic material, and the table also states that whether the type of hepatitis is chronic or not. So the hepatitis A and E is not chronic, but B, C, and D is chronic, and there is no particular care to either of the type of hepatitis. It's just that you tackle the infections and then you try to treat or comfort the patient. That is it. The hepatitis is mostly a fatal type of disorder which basically damages the liver severely, so that till to a point that the essential life-sustaining functions are even stopped. There is acute liver failure or hepatic encephalopathy, which is also seen, and liver cirrhosis or hepatocellular carcinoma, which is a liver cancer, could also develop due to hepatitis infection. Now there is dengue, which is a type of fever as well, which is caused by a virus. It is the virus here, which causes dengue, is also the RNA type of virus. It belongs to the Flaviviridae family, and the vector which transfers the dengue is actually the Aedes mosquito. When we get bitten by this mosquito, we actually acquire dengue. So it is basically classified under viruses causing hemorrhagic fever, hemorrhagic type of fever. The symptoms, it could include fever, headache, muscle, and joint pain, the usual fever symptoms. And in severe conditions, the hemorrhagic conditions, it can progress dengue to hemorrhagic fever as well or to dengue shock as well. So you usually notice the very prominent sign of this is you usually notice fever with a rash, which is a characteristic symptom of dengue, and it is also accompanied by break bone fever, which is due to like the pain you feel in the joints and the muscles. So first, what happens is the mosquito, it takes the blood from an infected person, then it bites. When it bites a healthy person, the dengue virus is delivered to the healthy person. So this hits the first wave of dengue, and it damages and the bitten site of the host body, which actually allows the dengue virus to come in. It activates the local mast cells there, the area which has been bitten by the mosquito, and macrophages and local dendritic cells also come there. Then after that, the second wave of dengue hits, which recruits the natural killer cells and the monocytes. The dendritic cells are recruited from the peripheral blood to tissue. The monocytes differentiate into inflammatory macrophages and monocyte-derived dendritic cells in the tissue. Then the activated dengue infected dendritic cells, they migrate, and it activates the adaptive immunity. The virus infected cells and circulating virons, they travel to the distant organs and they start their infection there also. They could travel to liver, spleen, lymph nodes, etc. So when they travel to distant organs, the virions of the dengue virus, they actually mature in the Golgi apparatus, and the mature virions are then released by exocytosis to infect further new cells as well. The diagnosis includes the evaluation of symptoms and serological tests to test IgM, IgG antibodies, or the PCR test to detect the viral RNA. The treatment is not very specific. It's just to manage the and relieve the symptoms and to intake a healthy diet and fluids, energetics, etc. And it is said to avoid aspirin and NSAIDs due to risk of bleeding and complications. Now we have the very famous coronavirus, which caused the global pandemic. So coronavirus is an enveloped virus. It is also an RNA virus, and it primarily infects the human respiratory tract. We all know about this, and the infection it begins when the viral spike protein, which it has, it binds to the host cell receptors, such as the ACE2 receptors, which is followed by a membrane fusion on the endocytosis. After the viral entry, the viral RNA is released into the cytoplasm and directly translated to produce viral replicons. So these replicated proteins, they form a replication transcription complex that synthesizes the viral RNA. So then these new virons they assemble and they are released by exocytosis, and that's how they cause the infection. It depends upon the severity of the infection, whether it could be sometimes mild also, sometimes very life-threatening, and it could lead to other severe conditions like pneumonia due to the immune-mediated inflammation as well. So there is vaccine available. I think by now we all must have taken the vaccines. So there is Covaxin and Covishield. So we all know about it. So this is about your coronavirus. So now we have chickenpox, which is caused by varicella zoster virus. So this is a highly contagious disease. It is also caused by the virus, and the person develops extremely itchy and painful blisters all over the body. It is usually occurred in the childhood. Some may acquire it in childhood, some could also get it in adulthood. And it basically happens due to the varicella zoster virus. It belongs to the herpes virus family. The transmission is via the air droplets through any infected person or a direct contact with the skin lesions of the infected person. The symptoms are extremely painful lesions, or you can say the skin develops harsh, like pimple-like conditions, which is extremely painful. The skin is flaky. It could be small, itchy, fluid-filled blisters all over the body, the red spots. And the diagnosis includes basically to test the type of rash and Zank's smear. The culture of vesicular fluid is also done. The treatment is generally self-limited. It's based if the patient is contained into a singular space, the immune system is the person is served with basic medications, and he or she is advised not to go out to spread the disease and to avoid scratching or rubbing the itchy skin. So these are the basic symptoms which we are mostly aware about. So the virus infection is shown in this picture. It is very in detailed format. So have a look and read it out. You can download it from the notes and have a look at it. You can get the notes from our Telegram channel. Now we'll talk about mumps. So mumps is also caused by a virus. So it's known by the characteristics of puffy cheeks and swollen jaw, which is a result of swollen salivary glands. So the common symptoms would include fever, headache, muscle aches, the swollen face, etc. So these are the common symptoms, and some tips to prevent mumps, or the most important point here, which I would like to highlight, is that the vaccine which is available for mumps is called MMR. So it's not only for mumps, it's for measles, mumps, and rubella as well. It's a three-in-one vaccine. So sometimes question is asked about this vaccine as well. So this is about mumps. It's also caused due to a virus. Sometimes mumps could be dangerous if not treated well. So it could lead to deafness, meningitis, or encephalitis. So this is all about mumps and the whole virus portion. So now we'll cover the fungi and parasitic infections. So firstly, we'll start with the fungal infections. First is Candidiasis, which is basically a yeast infection. So if it is happening in the oral area, so it will cause white patches, redness or soreness. If it is happening in the vaginal area, it will cause itching and thick white discharge. So the causative agent is the Candida albicans. It is Candida albicans is the most common agent. Otherwise, Candida glabrata or Candida auris are also one amongst the causative agent of candidiasis. And there is treatment and other diagnostic and pathophysiology as well, which you can read it out and do give it some time to read all the fungal and parasitic infections like that. I'll just quickly tell you the points which are most important with exam perspective. Now, the second disease we have is the dermatophytosis, which is the ringworm. Ringworm is the common name, or taenia. So the signs and symptoms are like if it is happening on the skin, so the skin will be red, itchy, ring-shaped rash will be there. If it is happening in the scalp, so hair loss or scaly patches will be seen. If it is happening in the nails, so the nails will be thickened, discolored, and brittle in nature. The causative agent, which is very important for ringworm, is the trichophyton or microsporum or the epidermophyton species. Now there is the other disease which is Aspergillus. So the signs and symptoms like this is something as allergic in nature. So this causes hypersensitivity, and people who are hypoallergic to spores usually acquire this disease. And the symptoms could be asthma, like wheezing, coughing, etc. will be there. Sometimes in severe cases, if it is aspergilloma, coughing up of blood is also there. And the causative agent is Aspergillus fumigatus or Aspergillus flavus. The diagnosis is usually done by a chest CT scan or galactomannan antigen test, or there is biopsy as well done. The next disease that we have is histoplasmosis. The causative agent is the Histoplasma capsulatum. The diagnosis, the mostly commonly signs and symptoms, if it is acute, so it will be flu-like. If it is chronic, then it resembles mostly TB, but most of the cases are asymptomatic as well. The diagnosis is usually done of the urine, or the serum could also be analyzed for antigen detection, or there is culturing or biopsy test as well. Then we have mucormycosis, which is also the black fungus. So the causative agent is mucor or rhizopus. The common signs and symptoms could be facial pain, black necrotic nasal crust, eye swelling, vision loss, etc. Then we have cryptococcosis. The causative agent is Cryptococcus neoformans or Cryptococcus gattai. The diagnosis, the signs and symptoms are the cough and the fever. It could also lead to meningitis. The diagnosis is India ink stain, so the stain is important. Now, there are some antifungal drug classes along with their examples and mechanism of actions. So you have to read it out, this column. You can get it from our notes, which you will find it on our Telegram channel. So I'll read out only one, which is the polyenes. This is the antifungal type of drug. And the examples under this is amphotericin and bemestin, which interacts, this the mechanism of action is it interacts with the sterols in the cell membrane, forming a channel that leaks cellular content. So this is how it like the action is done in the human body, whoever the person is taking this drug. So there are other drugs as well. So mainly you can remember, try to remember the drug classes and their examples. Now we come to parasitic infections. The first one is Leishmaniasis. The symptom includes irregular fever, anemia, weight loss, etc. The Leishmaniasis has three forms. So the first one is the visceral one, which is Kala-azar, which is the most serious one. Then is the cutaneous one, which is the most common one. And then we have mucocutaneous one, which so it all depends upon its type, and the fertility also depends on it. So it is very frequently asked the causative agent of leishmaniasis or Kala-azar. So the causative agent is something that you have to remember. So the causative agent of leishmaniasis is very easy to remember. It is caused by leishmania. It could have different stains like this Leishmania, major Leishmania, tropical Leishmania, Brazilianus, but Leishmania is the main causative agent of Leishmaniasis or even Kala-azar. Then we have malaria. So malaria we have all heard of. So malaria is a type of fever which is caused by a protozoan parasite. So the causative agent of mosquito, like the vector of this is the female Anopheles mosquito. It is transmitted by the bite of the female Anopheles mosquito in many tropical or subtropical regions. India has reported the highest number of malarial cases along with the second highest number of deaths due to malaria. Now we look at the pathophysiology of malaria. So first we'll start with where we get the transmission. So first, a healthy host is bitten by the female Anopheles mosquito. So the infected female Anopheles mosquito, it injects these sporozoites into the human bloodstream. Then these sporozoites, they travel to the liver, where they infect the hepatocytes, the liver cells, and they multiply there asexually, forming the merozoites. So some of the parasites here become dormant. They could form hypnozoites. causing relapse, like they won't further like actually form malaria. But the others, they develop and they are like the proper merozoites and they are released into the bloodstream. Once they are released into the bloodstream, they invade the red blood cells and they undergo asexual replication there as well. So the infected RBCs, they rupture, they release merozoites and a toxic metabolite which is called hemozoin. Hemozoin is very important. Hemozoin is the one metabolite which is toxic in nature and it actually is the reason why you get fever, chills, sweating and etc. of the symptoms during malaria. So the infected RBCs, they adhere and they adhere to the capillary endothelium and cause microvascular obstruction leading to cerebral malaria or organ dysfunction or hypoxia. So this is the life cycle of malaria. Some important points I would like to repeat again is that um, at the liver the parasite is asymptomatic. You don't really show the symptoms of malaria. Then at the RBCs it becomes symptomatic due to the release of hemozoin. You get the fever and chills. This is the kind of endotoxin that is released due to the rupture of merozoites. Then you see hemolysis, which leads to anemia basically. And the hypnozoites are the dormant form of the parasites, wherein you don't show like the malaria further does not aggravates. So these are some treatment goals of malaria, how you should prevent the further risk of attaining or other people getting. When you have some major anti-malarial drugs, do have a look at some of it at least, like your chloroquinone, amidoquinone, piperacquine. So the main suffix here is the queen word, which you can try, and this will help you to identify the antimalarial drugs. So there is queen suffix, there is INE suffix. So these are the most important ones. Now we have another disease by the protozoa called amoebiasis, which this is the amoebic dysentery, wherein the stool is basically diagnosed. So the transmission is through fecal-oral route and it leads to bloody diarrhea as well. Then we have lymphatic filariasis, which is also called elephantiasis, and the causative agent is very important here. The Bushraria banqueti is the most common one, and the Brugia malai is also the causative agent here. Then we have African sleeping sickness, which is caused by the Trypanosoma brucei in the West Africa chronic, or the Trypanosoma brucei rhodosidae, which is happening in the East Africa. So these protozoan diseases are very important along with their causative agents, very, very frequently asked in all the JRF and even GATE examination. So this was it for this video. I hope this rapid revision called all the kind of infections, bacterial, viral, fungal, protozoan may help you. And this was our best attempt to help you give a one-shot video for this unit. So don't miss this unit out. This is very important as well for your ICMR JRF examination and even for your other JRF examinations as well. So if you like the video, make sure that you share it with your friends, help them also study for their ICMR examination, and do give it a thumbs up. Comment under the video, and if you want any help that you want to us to do before the ICMR JRF examination, you can write it down. And make sure that you follow all the complete Part B rapid revision series with us. All the links for the videos has been there in the description box. Make sure that you watch those videos, and you could also join our Telegram groups. One is for the life science JRF notes and the other is for the updates of internship jobs and workshop opportunities. So you have the QR for both, and make sure that you subscribe to our channel and hit the bell icon so that you don't miss out on any notification from our side. And happy learning.